The unique properties of the atom-thick sheet of carbon, known as graphene, enabled a new penny-sized multiplexed biosensor that is the first to detect opioid byproducts in wastewater. A team of researchers from Boston College, Boston University, and Enid Labs report in the latest online edition of the journal ACS Nano. The novel device is the first to use graphene-based field effect transistors to detect 4 different synthetic and natural opioids at once, while shielding them from wastewater's harsh elements. When a specific opioid metabolite attaches to a molecular probe on the graphene, it changes the electrical charge on the graphene. These signals are easily read electronically for each probe attached to the device. The researcher says that this new sensor is able to rapidly, cheaply, and easily measure opioids in wastewater. Its sensitivity and portability would allow for wastewater-based epidemiology at the local scale, as specific as block by block or dorm by dorm, while ensuring privacy. The device responds to a primary challenge of the opioid epidemic, that is, determining the amount and kind of drugs being used in a community. Privacy concerns and limited resources are barriers to testing large populations. An alternative approach is wastewater-based epidemiology, similar to testing wastewater to measure community levels of coronavirus infection during the pandemic. Wastewater testing is an emerging strategy that can defeat limitations and stigma associated with individual drug testing, and it provides a more objective measure of drug use at neighborhood level. While wastewater testing has been widely conducted in Europe, only a few studies exist in the US. Rapid and portable nature of the team's device would allow wide-scale population testing at low cost and high geographical resolution. The team's prototype could provide a cheaper and faster tool for use by public health officials trying to determine the level of opioid usage and the impact of community-wide treatment interventions. While graphene has been used before for sensing biological samples, the team's work is the first demonstration that the material could be used with wastewater. In addition, it is the first demonstration of using graphene-based field effect transistors, an electronic device to read the amount of charge to detect multiple targets at the same time. The breakthrough was enabled by the design and implementation of the graphene electronic multiplexed sensor GEMS platform. The platform enables the sensing of 4 different target molecules at once, while shielding them from harsh elements in wastewater, samples of which were provided by the Mass Alternative Septic System Test Center on Cape Cod. The team fitted the graphene probes with aptamers, strands of DNA designed to only attach to a specific molecule, in this case metabolites of various opioids in wastewater. When the aptamer attaches to the drug, it folds, bringing more charge to the graphene. The amount of charge on the graphene is monitored to detect the presence of a specific opioid metabolite. Prior sampling tools faced the limitations because they record the shipping of samples and testing in a laboratory setting. Those requirements impose costs that limit wide adoption and use in communities without sufficient resources. By overcoming those limits, the graphene device can provide nearly real-time monitoring in multiple locations, which could also help distribute resources such as first responders or specific intervention strategies.